Hello, IB students. If you're here, you're probably on the struggle bus. You're probably struggling with understanding this topic. But fear no more, because I'm going to explain this so that you remember this for the rest of your life, or at least until exams. Let's try for a week at least. Okay. So first, we're going to go through everything very, very simply. The basis of inheritance. The basis of inheritance, okay, we're talking in sexual reproduction, is that parents pass genes to offspring, obviously, right? And for that, the gametes need to be haploid. So both the sperm and the ovules need to be haploid. Why? So that when they fuse together, it's diploid. Again, obvious, right? So the parents need, in the first place, meiosis to happen to produce the gametes. So far, so good, right? Next, this slide is kind of irrelevant, actually. I don't even know why I made it, but basically, it's just referring to the fact that to look at genetic crosses, to look at how genes get passed on, you can use flowers. This is what Mendel did using pea plants, right? He looked at tall versus short pea plants, etc. So it's very useful to reveal inheritance ratios, especially because plants do this process very quickly. Whereas if you want to look in humans, right, it's 9 months of pregnancy, etc. So that's the basics. Just also know that in flowers, there's male and female gametes. But actually, a flower can have both, so it can self-pollinate, but we'll talk about that later. So, genotype versus phenotype. Here's where it actually gets important, okay? What is the genotype? The genotype is simply the combination of alleles an organism inherits, okay? What are alleles? They're different versions of a gene, right? So, they're the same gene, but they're two different versions, okay? And that's what makes us different. In a way, right? We all have the gene for eye color. I mean, in reality, there's many genes, but you get the gist. But we have different alleles, okay? And we have 2 alleles per gene, one from our dad, one from our mom. Now, if we have the same allele for a given gene, then we call it homozygous. If we have different alleles, we call it heterozygous. So far, so good. Now, the phenotype are the actual observable traits. Traits are normally genetically determined by the genotype, but they're also environmentally influenced, right? And this is what's called phenotypic plasticity, this beautiful word here, which is the ability of an organism to change traits in response to its environment without changing its DNA, meaning without changing the actual genotype. And that's done through changes in gene expression. A good example is tanning, right? From increased melanin. These changes can be reversible, such as tanning, or permanent. Okay, good. Moving on. Alleles can be dominant or recessive. Basically, dominant alleles show with only one copy, so even if we just have one copy, the effect shows. Recessive do not. They only show when there's 2 copies, okay? So in this case, big T is the allele, this version is tall. Little t, it's dwarf, right? So the gene is for height, and there's 2 versions. As long as we have one big T, the plant is tall because the big T is dominant, right? So it's only when there's 2 little t's that the plant is a dwarf. Again, simple enough. Now, this, you would think you wouldn't have to remember this, but sadly you do. It was in the specimen paper. I assume it'll come up again when you least expect it. So what is phenylketonuria? It hurts to say it. It's a genetic disease, okay? And it is recessive. It's caused by a recessive allele. Why? Let's look at it. Okay, the gene we're looking at codes for this, for phenylalanine hydroxylase, which is an enzyme that converts phenylalanine to tyrosine, okay? So if one allele is dysfunctional, it's fine, because we still can produce some phenylalanine hydroxylase, so we're good. But when both alleles are mutated, meaning the non-functional one, we don't produce any enzyme that works, right? So no conversion happens, yeah? And basically, that's really bad because when phenylalanine builds up and it's not converted to tyrosine, it can lead to brain damage, basically, okay? So the issue with this is the parents might both be carriers, meaning they are heterozygous, but they don't have any of the symptoms. But then if they both pass the recessive allele, then the kid will have phenylketonuria, also known as PKU. Okay, and prevention for this is people who have it can just consume a low phenylalanine diet so that, again, you don't have too much, which would be bad. Okie dokie. Next thing, SNPs. Don't worry about this diagram yet. It's in the next slide. So SNPs, right, are single nucleotide polymorphisms. They're pronounced SNPs, actually, not SNPs. But they basically just means that there's a single base change in a gene. But that creates a new allele, right? So that's why in most genes, actually, we have multiple alleles, because as soon as there's an SNP, you get a new allele. And the gene pool is the collection of all the alleles in a given population. But remember, in the population, there might be tons, but a given individual, so like you or me, can only inherit 2. Okay? A good example of this is blood groups. Okay, so blood groups, there's 3 alleles. There's allele A, allele B, and allele, we call it little I, but like you can think of it as O. And basically they form 4 blood groups together. So when you have one A allele, you have the A antigen. When you have one B allele, you have the B antigen. If you have both alleles, the A and B, you have both A and B and you're type B. And if you have neither the A or B allele, so you have the little I or OO, then you're group O, okay? So what does that mean? It means that A and B, so these 2 alleles, are codominant. What does that mean? It means that one isn't dominant against the other. They're both dominant, right? Like, even though there's only one, so you have A and B, right? So you only have one A and one B, but both show up. Whereas, for example, little I, right? It doesn't show up unless you have the 2 little I's, because otherwise you're another blood group. I don't know if that makes sense. Any questions, leave it in the comments. And then the alternative to that is incomplete dominance, which is kind of different, but also kind of the same. The difference is in incomplete dominance, you have 2 alleles, right? So like big R, small r. But in this case, what happens is instead of showing both at the same time, such as here, so this is an example of codominance, you show both the A and B allele, your group AB. Here you don't show red and white, right? That would be codominance. If your petals were red and white, that would be codominance. But instead you show pink. That's somewhere in the middle, right? We call that incomplete dominance because the red is not fully dominant, right? It gets diluted by the white. Good. Next thing, sex determination. I think everyone knows this. I sure hope so. So humans have 23 chromosomes, right? Well, 23 pairs. And the 23rd pair are the sex chromosomes. So if you have 2 X chromosomes, you're a female. If you have 1 X and 1 Y, you're a male. Easy enough, right? Basically, the Y chromosome carries a gene called SRY, and that triggers the development of all the male features. If you don't have the Y, they just become ovaries. So actually, it's the sperm that determines the sex, because all eggs carry an X, but sperm carries either X, seen here, or Y. Good. So what happens when a gene is it's normally in the X, okay? It can be in the Y, but that's really rare. So when we talk about sex-linked inheritance, we're normally talking about about a gene being in the X chromosome. Okay. Actually, there's a lot of diseases that are sex-linked, meaning they're in the X chromosome. And by the way, they're almost always recessive, okay? So like, again, don't quote me on this, but like 95% of the times, they're X-linked and they're recessive. And what this means is that a male, as soon as they have one of the abnormal alleles, in this case, the little h, right, they're going to have the disease because they don't have another X to counterbalance it. Even if this little h is recessive, it doesn't matter because it's the only X chromosome you have, right? So you already have the disease. So a male is either normal, meaning they don't have the disease, or they have the disease. Whereas a female, right, can be normal, meaning she has 2 normal alleles. Then she can be a carrier as well, so big H, little h, or she can have the disease if she has little h, little h. But basically, these diseases are more common in men because as soon as they have one allele, they have the disease. Whereas for women, they need to have the 2 affected alleles. And an example of this, I think I already said this, is hemophilia, okay, which affects blood clotting. Okay, next thing, pedigree charts. So this is actually really hard to just teach theoretically. I would just recommend going and doing a bunch of practice questions. But just so you know, the squares are for males, the circles are for females, and then normally they'll change the color depending on whether they're affected or not. My one big tip for this, okay? Normally the question will go as follows. They'll show you a pedigree chart and it's a multiple choice. And they'll be like, what type of disease is this, okay? Most of my students, when we go through questions like this, they just jump to one. They're like, I really think it's recessive. Okay. They're almost always wrong. What you need to do is you need to go through every single of the 4 options and make sure that the one you're crossing out is impossible, okay? What do I mean by that? Here, you look at the affected, okay? And you look at their parents and you determine whether it's possible or impossible, okay? So a good example of this is, let's look at these kids, okay? Let's say that option A for the multiple choice is that it's a dominant allele, okay? Is that possible? Well, in fact, here it is, because this male would have a little a, let's call the gene A, right? It would have little a, and the mom definitely has a big A because it's dominant, but we don't know whether she has a big A, another big A, so she's homozygous, or a little a, right? And therefore, the kids can either be affected or not. So that's a possibility, right? Could it be recessive? Well, the dad would have to have a big A in that case, right? But the other one could be a little a, and the mom would have 2 little a. So again, that's possible. So from this, we can't deduce. But that's what I'm referring to. Sorry, I don't want to bore you with 5 minutes of this. But what I'm saying is, like, try and make sure you can rule out the option before you cross it out in multiple choice. That's all I'm saying. Next thing, continuous variation. So traits can either be discrete. That's when they have clear categories, right? Like blood type. It's AB, or O. It's not like O minus 2, you know? Or they can be continuous, where there's a range of values. For example, skin color ranges, right? And so continuous variation is normally caused by polygenic inheritance, meaning many genes are involved, and also it's often influenced by the environment. A good example of that is skin color, again. Okay, this I'm not even going to bother. I hope you know what a box and whisker plot is. If not, go to the math textbook. Okay, moving on to HL, where the good stuff always is. So, unlinked genes. Here, by the way, tons of students have confusion with this, but it's because the textbooks explain this horribly, okay? When we're talking about linked, unlinked, now we're not talking about one gene. We're talking about 2 or more genes, just to make that clear. And we're talking about how they separate together, right? We're talking about meiosis particularly. And we're talking about how these 2 genes separate during meiosis. So when genes are unlinked, okay, they are segregated independently of one another. For example, this gene and this gene are on different chromosomes, meaning in meiosis, they're going to separate independently, right? Where one goes doesn't determine where the other goes because they're on different chromosomes, okay? I hope that's clear. And I think that's simple enough because remember, this is an important concept in meiosis. There's independent assortment, meaning that like they can go wherever they want. They pair up as bivalents, right? 2 pairs of chromosomes, but then this one can go here, but it could go there, right? It's not preset where each one goes in which direction. So what I'm saying is that for unlinked, because they're on different chromosomes or really far apart on the same chromosome, what happens is that they segregate independently. Okay. Now, when you do a dihybrid cross, okay, a dihybrid cross is this. So like 2 heterozygotes for the 2 genes, right? So like big R, small r, big Y, small I, Y. So like you have your heterozygous for both, and both individuals that are being crossed are, okay? You have an expected ratio of 9:3:1. I really recommend you memorize this. That's all you need to know. In linked genes, that's not the case. Why? Linked genes are genes that are on the same chromosome, okay? So now this independent assortment stuff doesn't apply. Why? Because if one chromosome goes one way, it takes the 2 genes together, okay? I really recommend you just draw this, like it really helps. But because they're on the same chromosome, where one chromosome goes, the 2 genes go, okay? So now we don't get these ratios, and that's all you need to know. And all questions in IB are so predictable with this. They're all literally, we did a crossover between 2 heterozygotes for these 2 genes, and the ratios were 6:1:5:4. Are these linked or unlinked? And obviously, they're linked. Okay. So I understand why this causes confusion, because it's quite difficult. That being said, like, don't overcomplicate it, okay? So yeah, these linked genes are going to have fewer recombinants, right? They're going to have fewer combinations. I can go into more detail on this in the comments if anyone wants to, but for the sake of timing, I'm going to move on. Good. Next thing. Once again, I don't know why IB is deciding to just put random topics where they shouldn't be, but whatever. So, like, I don't even know what to say about this. You hopefully know there's the null and the alternative hypothesis. You're going to use this in your IA. That's all. Okay, so questions. A woman is a carrier for hemophilia and her partner has normal blood clotting. What is the probability that their son will have hemophilia? Okay, think about it. I don't want to wait, so I'm going to count down from 3. You can pause now, and then we'll show the answer. So 3, 2, and 1. 0.5. Why? Remember, hemophilia is sex-linked, okay? So the dad is going to pass on the Y, because we're talking about a son, not a daughter, okay? So the dad is going to pass on a Y. And then a woman is a carrier, meaning she can pass either the affected allele or the unaffected allele, 50-50. So there's a 50% chance that the son has hemophilia, okay? Because it's a son. Next question. A test cross involving 2 traits gives the following offspring numbers. 46 smooth long, 4 smooth short, 5 wrinkled long, 45 wrinkled short. What does this suggest about the genes involved? 3, 2, and 1. Okay, I'm actually so glad that I went over this question. Because, okay, you know how I mentioned that 9331 is for the dihybrid crosses. So when you have 2 heterozygotes for 2 genes, okay. Well, for a test cross, it's slightly different, okay? If you do a test cross, this is something you can just memorize, but if you do a test cross, what you expect is to get 1 to 1 to 1 to 1. That's for unlinked. So here, obviously, we're not getting that. We're getting 46 to 45, which is almost 1 to 1, but then we're getting 4 and 5. What is that about? That's because these genes are linked, basically, right? So these are the recombinants, but they're in much smaller quantities, so that's why they're linked. Okay. And last question. If a person with blood group A has children with someone who has blood group B, what are the possible blood groups of their children? Okay, again, 3, 2, and 1. I love this question because everyone always gets it wrong, and I love going through it. So, okay, theoretically, let's look at this. So the person with blood group A could have the A allele. Yeah, we spell it like this, I don't know why. The little I allele, and same thing for the B person. They could have the B allele and the little I, meaning they're both heterozygous, right? But they're still blood group A and blood group B. And this would mean actually that this person, their offspring could have A and B, meaning blood group AB, A and little I, meaning blood group A, little I and B, meaning blood group B, but also little I and little I, meaning O. So there you go. I don't know if you guessed this right. You've got a good hold of this topic, so congrats. That's it. That's a wrap. I'll see you next week for the next topic. Any questions, drop them in the comments. Bye-bye.